Let's jump right in. Welcome to This Explainer. We literally see medical headlines every single day claiming some kind of massive new breakthrough, right? But how do we actually know if a treatment works? Well, today, we're going to completely demystify the complex world of clinical trials. We'll explore the absolute gold standard of medical evidence, and we're going to do it by analyzing one of the most pressing, dangerous health crises of our time.
To understand why getting the science right matters so much, we have to look at the scale of the threat. The rate of doctor-diagnosed diabetes in England jumped from 2% in 1994 to 7% by 2019. Now, that's not just a slight increase. Diabetes is an emerging, full-blown global pandemic, and as our sources point out, this surge is being driven by a combination of an aging population, rising levels of obesity, and increasing physical inactivity. It's a massive, massive burden on global health.
Okay, so here's our game plan for This Explainer. We're going to cover the global epidemic, the anatomy of experimental studies, the sheer power of randomization, how we analyze trials using intention to treat, decoding those tricky trial statistics, and finally, how we balance benefit and harm.
Part 1. The Global Epidemic.
So the crucial point here is that medical interventions don't just happen at one stage. It's not a one-size-fits-all thing. They actually happen across 3 distinct levels of prevention. Primary prevention is all about stopping the disease before it even starts. Think modifying diet or increasing physical activity. Then secondary prevention kicks in. That's early diagnosis and weight loss to reverse or halt the progression. And finally, tertiary prevention. This is for when the disease is unfortunately already established, and our main goal is to prevent severe, life-threatening complications like cardiovascular disease.
Part 2. Anatomy of experimental studies.
Obviously, to know if these prevention methods actually work, we can't just guess. We rely on rigorous, heavily scrutinized experimental studies, and this brings us to the ultimate gold standard of evidence, the randomized controlled trial, or RCT. In an RCT, every single participant has an exact, equal chance of being sorted into either the intervention group, getting the fancy new treatment, or the control group, receiving standard care or a placebo. Pure chance and chance alone dictates who goes where.
Now, we design these highly controlled trials to answer very specific questions without letting human bias muddy the waters. Basically, we want to know 2 things. How effective is this treatment? And, just as importantly, who is actually most likely to benefit from it? Is this something for the general public or a very specific high-risk group?
Part 3. The Power of Randomization.
You can think of randomization as a medical detective's absolute best tool for hunting the truth. Why? Because it completely eliminates what we call confounding variables. By assigning people completely at random, it ensures both the treatment and control groups are mathematically comparable across all characteristics. I'm talking about the traits we know about and the hidden ones we don't. This massively reduces bias and allows us to comfortably blind the patients and the doctors so literally nobody knows who is getting the real pill. Ultimately, it gives us confidence that if we see a result, it was the treatment that caused it, not some mysterious hidden factor.
And honestly, this brilliantly illustrates randomization in action. This is from the Finnish Diabetes Prevention Study. They took middle-aged, overweight adults with impaired glucose tolerance, so a very high-risk group, and randomly assigned them to either individualized lifestyle counseling or a basic control group. Just look at the expanding gap between those outcomes over time. The intervention group achieved a massive 58% relative risk reduction in developing diabetes. Because of randomization, they proved that the lifestyle changes directly caused that drop. No doubts about it.
Part 4. Analyzing Trials with Intention to Treat.
Okay, but here's the reality check. Trials take years, and human beings are, well, completely unpredictable. What happens when real life gets in the way? Patients might experience really nasty side effects, find the new routine too difficult to maintain, or just drop out entirely. This is the single biggest threat to our trial blueprint. Think about it. If the absolute sickest patients drop out of the new treatment group because it's too hard on them, doesn't that artificially make the treatment look way better than it actually is?
The genius solution to this massive problem is something called intention to treat, or ITT analysis. This is absolutely crucial to understand. ITT means we carry out the final mathematical analysis based entirely on the group the participant was originally assigned to. It completely ignores what they actually ended up receiving. So, if you were randomly put in the treatment group on day 1, you were analyzed in the treatment group on the last day, even if you never swallowed a single pill.
Now, you might be wondering, actually wait, doesn't that water down the final results? Well, yes, ITT might underestimate how well a drug works perfectly in a vacuum, but here's the clincher.
breaking randomization ruins the entire foundation of evidence. If we just toss out the dropouts, we invite bias right back in. So ITT provides a completely unbiased estimate of the intervention's real-world effect. It preserves the absolute integrity of the whole operation.
Part 5. Decoding trial statistics. So, now that we know how these trials are built and mathematically protected, let's decode how the results are actually presented to us, the public. To understand the math, we're going to use a famous study called the CARDS trial. This was a tertiary prevention trial. They took patients who already had type 2 diabetes but no previous cardiovascular disease and randomly assigned them to receive either a placebo or 10 milligrams of atorvastatin, which is a cholesterol-lowering statin. The goal? See if the statin prevented heart events.
Let's look at the numbers. After 4 years, the results showed that on the placebo, 77 out of 1,410 patients had a coronary event. That gives us an absolute risk of 5.5%. But on the statin, 51 out of 1,417 had an event, making the absolute risk 3.6%. Keep those two numbers in your head, 5.5% and 3.6%.
Now notice the crazy stark contrast in how we can report those exact same numbers. If you divide the statin risk by the placebo risk, you get a relative risk reduction of 34%. And let me tell you, headlines absolutely love this number. You'll see, new drug reduces heart risk by 34% splashed everywhere. But look at the absolute risk reduction, the actual tangible numerical drop in real-world risk for the patient. That is just 5.5% minus 3.6%, an absolute drop of only 1.9%. Quite the difference, right?
Because the absolute drop is 1.9%, it leads us to a magical, game-changing number in medicine, 53. This is our NNT, the number needed to treat. You simply divide 1 by that absolute risk reduction of 1.9%. What does it mean? It tells us that we must treat exactly 53 people with statins for 4 whole years to prevent just one single coronary event. The other 52 people take the drug every day, but it literally doesn't change their outcome.
Part 6. Balancing benefit and harm.
Realizing that we have to treat 53 people just to help one brings us to our final, critical evaluation. All medical treatments, no matter what they are evaluated by carefully balancing these benefits against potential harm. We have to constantly ask, was the trial large enough that this wasn't just a fluke? Did they use strong blinding and that intention to treat analysis we talked about? And crucially, are the results actually generalizable? Like if a trial only studied healthy middle-aged men, we definitely can't just assume the drug works exactly the same way for elderly women.
And just like we calculate the number needed to treat to see a benefit, we absolutely 100% must calculate the number needed to harm, or NNH. This tells us the average number of people who need to take the medication for one person to suffer a severe adverse event. For instance, in diabetes trials that push for really intensive blood sugar control, we have to weigh the benefits against the massive risk of severe hypoglycemia. Basically, if your number needed to harm is lower than your number needed to treat, that intervention is going to cause a lot more problems than it solves.
So, what does this all mean for us? I'm going to leave you with this. The next time you see a flashy clickbait medical breakthrough in the news, what are you going to ask yourself? Remember that only high-quality randomized controlled trials deserve their place at the top of the evidence hierarchy. Stay curious, stay highly critical, demand to know if the trial analyzed everyone who started it, and always check the absolute math. Thanks so much for joining me on this explainer. See you next time.